Hey everybody, it's Will from the Micro Lab. Today I was going to try to start a series about how one can make petri plates out of just common household objects. I was looking to start and make these petri plates out of chicken broth today. It's a very simple setup where you can just take the chicken broth, mix it with agar, the substance that makes it sort of into a gelatin, pour it in these petri plates, and then my idea was to just go around the house and find fun places to collect environmental samples, be it from the dirt outside or my pet bunny's ears. Something exciting. And since, of course, this stuff is edible, maybe I'll end up licking one and see what can grow on the petri plates then. So with that, we can just jump right into it. First, I measured around 350 milliliters of the chicken broth, just using a standard household measuring cup here. Later, I'm going to add this to a mason jar. The mason jar in this case is autoclavable. I have a sort of canning setup that you could use for pickling or for canning fruits and vegetables. In my case, I mostly use it for autoclaving things. Next, I measured around 5.5 grams of the agar here. This is the stuff that makes our plate gelatin-like. In this case, I just use a small sauce dish and my mom's food weighing scale. Next, I combined the 350 milliliters of the chicken broth and the 5.5 grams of the agar powder together in a mason jar. I then closed the cap and lightly shook the contents to make sure everything was nice and distributed equally. Next, I went to go set up my autoclaving apparatus, which is just a Presto 24-quart canning pot. As the directions indicate, one should put at least 3 quarts of water in at the bottom. This water will create steam later once pressure is generated. The pressure and the high heat, again, will work to sterilize any media or anything that you put in there, fruits, vegetables, and in my case, my chicken broth, which I will later pour into plates. The chicken broth was set to autoclave for 30 minutes at around 15 psi. The temperature within the canner should be around 265 to 280 degrees Fahrenheit, which is plenty hot enough for sterilizing media like this. I went ahead and poured the plates. The volume of these plates are around 45 to 50 milliliters. Really, you can just eyeball this step. Really, the rule of thumb is that you can pour the plates such that it just creates one smooth layer. You don't want to add too little such that there's gaps in the agar. You don't want to add too much such that you can't pour other plates, basically. It's totally fine to have some agar solution left over in the end. It's important here that you also want to sterilize the surface, in this case my kitchen countertop. I just used some dilute bleach in this case. The Bunsen burner here actually acts as a way to generate a sort of as I was taught, a heat curtain that pushes away any aerosolized fungal spores or bacteria. Once I'm done pouring the plates, I also like to put the lid on slightly ajar. This makes it so that condensation from the hot agar solution doesn't rise into the glass and make it really foggy and wet. Once they're all poured in this case, I just put all of the lids on them and let them sit under the Bunsen burner for around 5 minutes, and then I put the lids back on and turn off the Bunsen burner. All in all, they don't take very long to cool and solidify, around maybe 10 minutes from the time you start to pour everything. And once that's done, we can go ahead and take some environmental samples. So using some sterile swabs, I went ahead and grabbed some environmental samples. I took one from the dirt, one from my nose, one from my phone. I can do this by just scraping each surface with these dry sterile Q-tips here. I also have a really cute pet bunny who's really nice at being handled. I took a sample from the inside of her ear just to see what could potentially grow on these chicken broth plates. I also have this mason jar full of pretty disgusting pond water. I figured this would be a pretty good control for what we potentially could see on these chicken broth plates. I bet you something would grow. So here are the results after incubating these plates in my garage for around 24 hours. The temperature in my garage is around 75 to around 85 degrees Fahrenheit, which is pretty much perfect in this case. Here is the dirt plate. As you can see, some of the dirt particulate at the top. I saw around 4 to 5 colony morphologies here, so that's 4 to 5 different bacteria that are growing, in this case, on the chicken broth here. Next came the plate from the pond water. As you can see, there's around 3 to 4 different colony morphologies easily seen on this camera. I personally counted around 4 to 5, so these are 4 to 5 different bacterial species that the chicken broth sort of suits them to grow on. I can almost guarantee you that there's dozens and dozens of other bacteria present in the pond water and even on the dirt, but the chicken broth only has the ability to let only certain types of bacteria live on it. In all of the other cases, the plate that I licked, the plate from my phone sample, the plate from my nose, and the plate from my bunny's ear, there were no colonies on it. which is kind of sad in this case for this video, but that just indicates that bacteria living in these places, which I can guarantee, cannot live or inhabit the chicken broth here. But again, that doesn't mean that there aren't any bacteria from those locations. It just means that the chicken broth was unable to support bacterial life in those cases. That pretty much brings us to the end of this video. In conclusion, we saw that samples from diverse environments, like the soil, like pond water, have bacteria that can grow on chicken broth, while other environments, like inside your nose, on your phone, don't have any bacteria that can grow here on the chicken broth. Again, this is hopefully going to be a nice series. I think the next one that I'm going to do is Coca-Cola, make that into plates and see what bacteria I can make grow on it. Hopefully I'll find something in the soil that can grow on those. But again, that's pretty much it. Thanks for watching. Feel free to subscribe. Feel free to leave a comment on anything. But as always, have a great rest of your day.